All right. Yeah, thanks so much for the invitation, and thanks so much for meeting with me today, guys. I had a really lovely day chatting with you all, and I hope I can finish off your day with an interesting talk. And I'm going to tell you about some of the work I and we've been doing to understand the tempo and mode of adaptation. Pretty classic concepts in evolutionary biology, but kind of translating that to think about how these relate to adaptation across contemporary landscapes, mostly in response to the agriculturalization of our landscape. So I think as biologists, we're all acutely aware of the impact that we as humans have had not only on shaping our environments, but the organisms that live in them, right? And I think some of our strongest examples of evolution come from these human-mediated selective pressures, right? We can look to, let's see if I can get this mouse tracking. We can look to industrialization in the 1900s, driving selection for these darker colored pepper moths, better camouflaged against the soot-covered trees, right? A very classic example of rapid evolution. Overfishing of Atlantic cod off the coast of Canada, driving rapid life history evolution, or even more recently, the evolution of tusklessness in African elephants as a result of the ivory trade. Right, so these are all examples of really rapid evolution in populations and species, but I think we're increasingly aware of the fact that this is unlikely to be the case for most populations and species in changing environments. So our lab is really interested in understanding what determines whether, but also how quickly populations can actually respond to environmental change. Right? And so we can turn to a framework really at the intersection of ecology and evolutionary biology to address this question, and that is evolutionary rescue theory, right? And so this posits that the intersection of these two types of processes, demographic change, no, okay, demographic change, right, which refers to changing population sizes, and the availability of adaptive genetic variation that determines the likelihood of a population's response to changing environments. So we can imagine after some environmental change here that happens at the x-axis equals 0, population might no longer be suited to the environment in which they're found, right? And so you undergo demographic collapse, reduction in population size, but if a new beneficial mutation can arise, this can rescue the population and allow them to undergo local adaptation or evolutionary rescue. But this is actually a pretty slow way in which populations evolve, right? Because there's all of this waiting time for these new beneficial mutations. On the other hand, in larger populations with larger effective population sizes, there should be more variation standing in those populations, and that might mean that it can facilitate a much more rapid response to changing environments. And indeed, since the seminal review and perhaps even before, we've seen many cases of standing genetic variation facilitating rapid adaptation across different populations and species. So we're really interested in kind of bringing this framework together to understand how plants evolve to changing environments, and mostly these really extreme human-mediated selective pressures. And so we bring in population genomics with spatial and temporal sampling to really understand how variation is structured at these different scales. We complement this with quantitative experiments to figure out how this variation translates to phenotypes and fitness in the wild. And then, of course, theoretical inference to understand what are the mechanisms in parameter space, which is really limiting the response to these and determining kind of the distribution of variation across our landscape. So when we talk about environmental change, I think one of the most striking changes we can look to is really the increase in conversion of our land for the production of food, right? Cultivation of crops. So of course you can see my North American centric figure here, but in the 1600s across North America, United States and Canada in particular, we were only using 1 million hectares of land towards food production, but at its maximum in the 1950s, over 10% of North America's land surface. Right? And of course, this expansion period really aligned with the human population growth, which took a large proportion of the workforce out of agriculture and into other industry. And while this met our world's growing population for quite a time, at some point we realized we could no longer convert land to agriculture. We had to figure out other ways to efficiently produce food. Hence came what some would call the Green Revolution, right? And so in the 1950s, it was actually an evolutionary innovation, the breeding of high-yield crop varieties that facilitated this increase in the efficacy of food that I would say really facilitated the intensification of selective pressures across the landscape for organisms experiencing these environments, right? And so this started with this evolutionary innovation, but along with it came highly controlled irrigation, widespread mechanization, and increase, and some would say, over on chemical inputs like pesticides, fertilizers, etc. And indeed, I think we see clear evidence of this intensification when we look at this unintended response, which is selection for herbicide resistance across species. So since herbicides were first introduced across the landscape in 1950, we've actually seen hundreds of species evolve resistance to every chemical compound that we've been able to innovate to lethally control weeds, which outcompete crops for resources, right? So these draw down crop yields. And because of the widespread nature of herbicide resistance, not only within but across species, this is a really economically important problem, right? And so in the United States alone, this is a much outdated figure, but this costs many billions of dollars per year in terms of both the effort that goes into managing these weeds, but also the drawdown on crops that these weeds result in. So as a result of its importance, there's actually been quite a lot of work to understand the causal mechanisms of herbicide resistance. So not only the genes targeted by these different chemical compounds, but actually that particular amino acids and non-synonymous mutations within them that result in resistance to herbicides across species. Right, so from an evolutionary perspective, I think this sets up a really pressing but also exciting system to address these questions, and pretty classic questions in evolutionary biology. That is, what is the strength of selection that are governing these responses, and how quickly does this occur across the relevant spatial and temporal scales? And really, what are the modes through which herbicide resistance is spreading, right? What is the role of new mutation and migration in the spread of resistance across our landscapes? So these is kind of the two parts of my talk today that I'm going to focus on. And so to set the stage a little bit more, I want to introduce you to a weed that maybe you're not so familiar with here in Austria, but if you were out in the Midwest in the United States, might be a typical picture. So this is actually a fallow field, so a farmer isn't actually cultivating any crops on this, but this field actually coexists nearby a soy field. And what you can see up there towering high above kind of the canopy in red is a common water hemp. And this is one of the most problematic agricultural weeds in the Midwestern United States, in part because of really high levels of herbicide resistance. But it's also dioecious, meaning it has separate sexes, it's wind pollinated, and it's diploid. So it's kind of this perfect storm, potentially really high levels of genetic variation, gene flow, and it also has super high levels of seed production. So very fecund. A single plant can produce up to 5 million seeds, right? So it's perhaps this worst storm for kind of rapidly evolving to human-mediated management. From an evolutionary perspective, it's also convenient because there's two native subspecies or varieties that structure genetic variation across the range. So we have some expectation about where we expect to see certain variants over the landscape. And so I've been developing the system to better understand rapid adaptation to herbicides, along with mentors and collaborators. We've developed genomic resources, including first reference genome for the species, collaboration with Detlef Weigel, and also weed scientists at University of Illinois near Chicago, and my PhD and postdoctoral mentors at UBC and University of Toronto. So kind of mirroring this question I set up, whether and how quickly populations respond to environmental change, I think we can use water hemp to really understand how quickly the species has really evolved to agricultural change. And so in order to do this, we can leverage the fact that this is a native species. This species is native to the Midwestern United States. So we can actually sample both the ancestral and derived habitats, right? And so, several years ago, I was able to do a road trip across the US and collect populations from both these derived invasive agricultural habitats, but also these natural riparian wet land habitats that water hemp still exists in, right? And we can use this to contrast what are the drivers of broad-scale genetic variation across the range versus consistent changes that have to occur in order for this plant to successfully invade agricultural settings. And for a little more context, here's just a picture of what these environments look like. So here in southern Illinois, on the top, we can see this is a soy field just a few weeks out from harvest, really overran and outcompeted by water hemp that's really towering over this landscape, outcompeting it for light and resources. But just a kilometer away or a couple kilometers away, a pretty sparse population of water hemp alongside this body of water occurring kind of in this sandy ditch next to the lake. So very different ecological and environmental selective pressures likely occurring in these habitats. So the first thing we might ask is there kind of the evolution of phenotypes that might relate to fitness, or is there local adaptation of populations collected across these different environments, right? And a pretty classic way to do that is a common garden experiment. I talked to many of you today about how these have been used in Drosophila, evolve, and resequence experiments. Here I took those populations from those 34 natural collected or wild collected populations and grew them all in a single environment. And actually, I say single, but what I actually imposed 2 treatments. Water supplementation treatment, an important part of these natural habitats, right, is that there's lots of access to water. We also did a treatment where we competed them with soy. That's another important part of agricultural habitats. And we also grew them up in a control treatment just in the pot alone. And the idea is that we could kind of do a twist on this reciprocal common garden experiment, which is the gold standard for local adaptation. Do plants or organisms do better in their home environments compared to away? And so what we expected to see was that agricultural plants might do better when grown in soy compared to plants from natural habitats, and natural source plants might do better than agricultural source plants when grown in these water supplemented treatments. And so we phenotyped for 12 traits across the life history of this plant, and we actually did whole genome sequencing of nearly 200 individuals. And so what I'm going to show you is these kind of home and away metrics across these different treatments, so water, control, and soy, with their source environment in different colors. And dry biomass here is a very important feature of plant fitness, right? Larger individuals produce more seeds. And so the thick line is the average across environment types. The thin line are the population level reaction norms. And what we're not seeing really is consistent crossing of these reaction norms. It's consistent kind of signal of antagonistic pleiotropy, where agricultural plants do better in soy and worse in water. We do see a significant shift towards larger plants in agriculture. And we also see a significant genetically determined shift towards later flowering plants in agriculture. So there's been a kind of a clear shift in genetic variation that's been selected on during this transition into agriculture. But from this phenotypes, we're not really seeing a clear link to local adaptation, or that agricultural plants are doing better consistently in these particular treatments. So while there's been clearly phenotypic evolution, we wanted to better understand how this might relate to signatures of selection. And so we came at this from another angle, and that was with genomic data. And so from these same collections, we sequenced about 200 individuals, and we were interested in what regions of the genome might underlie kind of this invasion into agriculture. So the first thing you might think to do, which is what we also thought to do, is to look at genetic differentiation across these natural and agricultural habitats. What was very striking to see is extremely low levels of FST, and I get this tracking. So FST value close to 0, meaning that most of the variants found in agricultural habitats are also found at the same frequencies in natural habitats. There's very low levels of divergence to the extent that it represents near panmixia or complete gene flow across these habitats. So this is really kind of important in and of itself, because it tells us that the selective regimes and management that we're applying in agricultural habitats are actually playing a really important role in shaping genetic variation in nearby natural habitats. But we can also leverage kind of genome-wide variation to figure out what are the regions that in particular are playing a much more important role in this response into agriculture or local adaptation. And so what I'm showing you here is a signature of this test. And so what we asked was whether there were SNPs in the genome that were only ever found in one environment or enriched in one environment consistently across this paired design, right? So this is a very similar statistical approach that is used quite often in evolve and resequence, this Cochrane-Mantel-Hansel test. And this p-value on the y-axis, a larger value indicates more evidence of playing a role in adaptation to agriculture. So despite this low genome-wide FST, what we actually find is numerous regions highlighted in red all across the genome that are significantly associated with adaptation to agriculture. These are enriched for functions in response to growth, reproduction, stress, and herbicides. And actually, the region of the genome with the highest evidence or the strongest evidence of adaptation to agriculture actually falls directly within this gene PPO, which is the target of PPO inhibiting herbicides. So perhaps not that much of a surprise that these lethal selective pressures leave some of the strongest selections of signature across the genome, but really kind of driving home the importance of herbicides in adaptation to agriculture across the landscape. So we're testing, we're taking this pair design across the two environments and asking, in each pair, are there SNPs that are consistently enriched in the same direction in agriculture compared to natural populations? So we're testing each SNP one at a time, but we're taking information across both environments and across all pairs all at once for each SNP. No, we are asking whether or not there's a SNP that's found at a certain frequency in the environment consistently across pairs. So whether it's found at a certain frequency depends on what environment it's in. Well, I think there's multiple forms of the Cochrane-Mantel test. We can ask whether the SNP effect depends on the pair, or we can ask whether SNP effect depends on the environment. So the test that we did here, we're asking whether there are SNPs that are consistently found in a certain environment across our pairs. Right, and so this statistic is what I'm showing is very similar to if we were to do an FST at each SNP in the genome, and it shows much. They're very highly correlated, these FST and CMH statistics. But this is leveraging the fact that our design is built on this paired replicated sampling. So I think not only can we use that data to identify particular regions of the genome that might be important, but it also gives us a suite of alleles that we can use to learn about the evolutionary history of adaptation to agriculture. And we can actually do that by leveraging time series data over the same kind of spatial scale as which we have these contemporary collections, right? And so I was able to curate a set of samples dating all the way back to 1828, working with 10 herbaria across North America, mostly the United States. And what was really exciting about this is that we could not only get this time series data dating back nearly 2 centuries, but we could also curate a set of samples that spanned kind of these different selective regimes. So because of the really well annotated labels on these museum specimens of these plants, we could determine whether they were collected from agricultural, natural, or disturbed habitats. And so even in this species, I'm not the first to think about kind of this tempo of evolution in water hemp and what might have facilitated its invasion into agriculture. This botanist in the 1950s, Jonathan Sawyer, actually used some of these very same herbarium specimens to test how kind of these two subspecies and range expansion, specifically of Var rudis, might have facilitated the invasion into agriculture. So based on morphology data in the herbarium, he actually hypothesized that Var rudis expanding northeastwards really facilitated a lot of the invasion into agriculture we're experiencing today. So the first thing we could actually do with this herbarium data is test Sawyer's hypothesis. We could reconstruct the demographic history of this species. And I meant to say that you can see that as opposed to rudis, this Var tuberculatus seems to really closely mimic the major Mississippi and Missouri water basins, which kind of reflect its kind of riparian associated nature. And Sawyer also noticed that in contrast to rudis, which seemed to be much more kind of disturbed adapted. So what I'm going to show you here is kind of the distribution of this Var rudis ancestry through space, across longitude on the x-axis, but also through time, right? And so I've binned kind of our herbarium data into 3 different time points, and each color reflects a different time point or time range. And so in our oldest time range from 1828 to 1920, we can see that there's a strong climb in ancestry. Just as Sawyer thought, to the west, we see nearly pure var rudis ancestry, and to the east, nearly pure var tuberculatus. But what you'll notice is that as we move into more recent timescales, up here in yellow, we can see there's been an expansion of var rudis ancestry eastwards across the landscape, just as Sawyer predicted from morphology data. But what's interesting, instead of plotting these individuals by what time frame they were collected in, we can actually look at what environment they were found in. And when we look at contemporary or historical data, we saw no difference in the distribution of ancestry across natural and agricultural habitats. But when we bring in our contemporary data, we can actually see that this expansion of var rudis ancestry eastward has really driven the excess sorting of that ancestry into agricultural environments. So we're seeing actually quite an excess, oh, we're really not tracking, an excess of var rudis ancestry in agricultural compared to natural habitats in a given place across the landscape. We can then ask how this relates to these fine scale signatures of adaptation across the genome, right? So what we might expect is that regions involved in adaptation to agriculture might come preferentially from kind of this var rudis haplotypic variation. And so when we look at regions of the genome that show that role in agricultural adaptation, so to the right on this y-axis, right, based on the CMH scan, we can see that those have the biggest excess of var rudis ancestry in agriculture. And so what this tells us is Sawyer was actually right, that standing genetic variation or ancestral variation in var rudis has played a really important role in facilitating a rapid response of this plant's invasion into agriculture. It also really fits with kind of evolutionary rescue theory, where standing variation might be really important for facilitating a rapid response over such short timescales. So we can use this type of time series data to reconstruct changing demography or the distribution of ancestry across the range. But I think what's also really exciting is we can use it to perform direct inference of the timescale of adaptation and the strength of selection from this historical sequence data. So I want to walk you through really briefly what this looks like. An important thing to keep in mind is that with this herbarium data, we're only able to sample a single plant at a single year in a single locality. And so each point here we can think of as an observation of an individual's genotype from that collection, spatiotemporal collection. Right? And so an individual is either homozygous recessive, heterozygous, or homozygous alternate. And if there was selection, we might see a signal like this, where maybe that allele is increased through time across samples. Well, we can just think about this conceptually or theoretically, how we might actually infer selection. So what we have here is an observation of an individual's allele frequency, for example, here at time t, or p at z, or sorry, p at 0, right? And we also know for other samples what year they were collected in, that's t. And so what that tells us is that if we have these observations across a time sequence, that we can actually rearrange that model to take an even more generalizable form, and that's just a logistic regression, so that we can actually reconstruct p at t, right? That's our predicted allele frequency through time. And the slope parameter of that logistic regression is actually just equivalent to the strength of selection, right? The relative fitness advantage that that allele confers across the scale of your collections. So now I can fit this relatively conceptually intuitive logistic model to understand the strength of selection governing adaptation to agriculture. And so I'll do this again for these alleles that we've already identified to play an important role in adaptation to agriculture in our contemporary samples, first focusing on samples collected from natural habitats in our herbarium data. So here are those allele frequency trajectories, and they're just colored by either how much they've increased in dark purple, meaning they've increased the most, or increased the least through time in yellow, some of which are actually decreasing, right? So over this time scale, the last 150 years, we see alleles have increased on average by 6%, But actually, over this time scale, it's not consistent with significant evidence of selection. Our S confidence intervals are overlapping 0. And if we do kind of permutations of genome-wide data, it actually even falls within the null expectation. On the other hand, if we can reconstruct the trajectory of these alleles sampled from agricultural plants in our herbarium data set, we can see that these alleles increase much more, 22% over the last 150 years, consistent with a strength of selection of about 1.5%, so relative fitness advantage of 1.5% over the last 150 years. So very strong selection over the spatiotemporal scales that we're looking at. And we can even use the same type of framework to ask to see if there's been evidence of a shift in selection through time, right? And so what we want to know is if we were to fit a single logistic or multiple logistic regressions, does that better explain kind of the variation in allele frequency we see? We might also be interested in asking where that shift might actually occur in our time series data. And so we can do that by fitting kind of a piecewise regression model. And the breakpoint in that data actually falls naturally out between this period of expansion and intensification, right in the 1950s, where these agriculturally or contemporary agriculturally adaptive alleles prior to the 1950s don't actually show significant evidence of selection when we break it out like this. But after the 1950s, we see an S over the last 40 years of closer to 5% on average in agricultural habitats. And we're even seeing evidence of selection in natural habitats, which might relate to some unaccounted for migration in some of these samples. But regardless, we're seeing really extreme strength of selection over extremely recent timescales in water hemp, driven by the intensification of agriculture. And I also want to bring your attention to the very high frequency of these alleles even before the onset of selection, right? So again, a very important role in standing variation in facilitating this really rapid response. Because we're very interested in these resistance alleles as well, right? These are causal resistance alleles that we know are important for fitness in the field. And so we can actually parameterize a maximum likelihood framework for inferring kind of the range of selection coefficients that might be consistent with the data we observe, right? And so these are 7 herbicide resistance alleles that are very common in our contemporary data. And so when we compare the frequencies of these alleles in our contemporary compared to herbarium data, we actually see a kind of a marked shift on extremely recent timescales. So for our most common allele, for example, this curve in yellow, the global contemporary allele frequency is about 25%, but we actually only see 2 historical samples that show any evidence of these resistance alleles. And to account for that large shift, there has to be extreme selection on recent timescales governing these resistance alleles, such that the upper limit of selection on this, our most common allele, is all the way up to 0.34, right? And so this is, extreme selection over this time scale, and I think probably even an underestimate considering that these are lethal selective pressures applied within the field. So this is probably capturing also a lot of the spatial and temporal variation in selection governing these alleles at the scales that we're looking. So in summary, we're seeing in terms of the strength and tempo of adaptation that despite pervasive gene flow, we're seeing evidence of polygenic adaptation through genes related to growth, reproduction, tolerance to stress and herbicides. That demographic change has played a really important role in this response, where the expansion of varrootist ancestry northeastwards has really provided the standing variation on which selection could act as it moves into agriculture. And that this response to selection has been much more rapid and extreme than is typically observed in wild populations, kind of reinforcing the strength of these human-mediated management and selective practices in shaping organisms across our landscapes. And so some of our ongoing work now is to ask about how these responses to selection vary across timescales, right? Farmers are performing these on-field selective experiments year after year at unprecedented scales. And so we've reached out and kind of made our own network of kind of this on-field evolve and resequence experiment where we're actually sampling multiple times within a single generation to see how quickly or how rapidly these populations are responding, both in terms of demographic response and response at these adaptive alleles, in response to multiple herbicide applications, in response to different focal crops or different environmental variables, to get at how consistent the changes we see are at different scales or different fields, understand the strength of selection and the dominance that are governing these alleles across these populations, and better address how much of the genome is actually responding to these on-field selective pressures. And so we've already done a year of collections, and this year we're going back to get a sense of how much continuity there is in the responses that we've seen. We're also interested in understanding why rudous ancestry was actually favored into agriculture. What pre-adapted it? And so one of the hypotheses that we had is that as this plants moved from riparian habitats into environments, agricultural environments that have less water availability, that drought adaptation might actually play an important role. And so we performed a drought experiment where we grew water hemp in the greenhouse, we stopped watering it, and we tracked survival. And what we actually see is evidence of climate adaptation, right? So southwestern populations that experience drier habitats survive longer. We also find, sorry, the legend isn't there, but those are those blue lines. We also find that agricultural populations in solid tend to be more drought tolerant than natural ones, especially in the middle of the range. And that actually relates to the distribution of ancestry across these populations. So more rudous ancestry leads to more drought tolerance in this experiment. And so we've done sequencing across the extent of this survival experiment. And we actually did admixture mapping, or a mapping of ancestry haplotypes to drought, to show that there's this polygenic response to drought. And that if we look at the allele frequency change of these alleles across the course of this experiment, that we can recover pretty extreme response to selection all across the genome, selection coefficients on the order of about 5% again, and that each of these alleles provides strong protective effects against drought. So hazard ratios from about 0.5 to 0.75, indicating about a 30% increase in the probability of survival having any one of these alleles. Apologies. So drought seems like has played an important role in shaping the genetic variation in Barbados ancestry, which might have also facilitated its response to agriculture. And then we're also looking to couple this again with our herbarium genomic time series to understand, the extent to which the tempo of adaptation within extreme selective bout might actually relate to the signatures of adaptation we see over much longer timescales with changing climate. I think I just have a couple minutes left, so I'll speed through this last section. It's just a couple more slides. But I talked a lot about, in my introduction, kind of expectations from evolutionary rescue theory that new mutation might actually limit the pace of adaptation. And so that's not really come to play out in any of the results I talked about so far, but I think there is an interesting angle that we could take to test this question. And that's to ask about the extent to which new mutation limits the pace of adaptation for these large effect alleles, right? So these herbicide resistance alleles on their own can confer sufficient resistance for these plants to survive in the face of herbicides. And these three mutations, two within ALS, one within PPO, are really common in our contemporary population. So it's a great framework for asking this question, how limiting new mutation is in the spread of resistance. And because these new mutations occur on separate haplotypes, we can actually reconstruct the evolutionary history of these mutations to get a sense of, indeed, whether there are multiple haplotypes on which these mutations arose. And so when we reconstruct these haplotype trees, what we can actually find is that based on the clumping of these colored tips, which represent a resistance mutation, that each of these mutations have actually had multiple independent origins on separate haplotypes, right? So clearly there's evidence of repeated origins of these common resistance conferring SNPs. And that doesn't really jibe with this hypothesis that new mutations are limiting. And so we wanted to try and resolve that potential conundrum. And we can just turn back to very simple or classic population genetic theory, this expectation that theta equals 4ne mu, and then when you divide by 2, we can actually interpret that parameter to be the mutational input at each allele every generation. So we know that the mutation rate is likely very low, but we can ask perhaps how changing effective population size might facilitate us being in this parameter space where mutation isn't actually limiting. So with genomic data, we can reconstruct the changing effective population size through time based on coalescent rates across the genome. And what you'll notice is that historically, there's been very constant effective population size from like 1 million years to the last 100 years. But when you look from 100 years into the current time, there's been a massive effective population size expansion by multiple more orders of magnitude, right? And so when we use this contemporary NE, which is all over the same time scale in which herbicides have been introduced, and we plug it into this equation, what we get is this theta greater than 1, and which tells us that somewhere across the genome, every generation, a new mutation is rising somewhere across the landscape, right? So if these populations are connected enough, then adaptation at a single site might not actually be limited by mutation. And so this is pretty striking. And in Drosophila, they've seen similar results where soft selective sweeps are facilitated by this very large contemporary effective population size. And bad news for managing resistance is not just repeated mutational input, but also gene flow that's spreading these resistance alleles across the landscape. So if we take these independent mutational origins and we map them back onto the populations on which they're found, we see that those mutations are found not only across multiple populations, but across multiple geographic regions, even across country borders. So up there in this little square, that's actually Canada, down here, Midwestern United States. But not only that, a single population might harbor multiple independent mutational origins of the exact same SNP, right? So both a widespread role of gene flow and new mutations in the spread of resistance. I'm going to skip that slide. And so in terms of the kind of nature of monogenic adaptation, I think we've also seen this role of demography feeding back and facilitating rapid adaptation, right? This changing effective population size, and that repeated origins in gene flow have really driven the spread of resistance. And I think this really fits in with the patterns we've seen when we talk about adaptation across the genome or at these particular sites, that these patterns of range expansion and gene flow, just like as expected from evolutionary rescue theory, have facilitated rapid adaptation through standing variation. That now that these populations are persisting in these habitats and kind of evolving to this new fitness landscape, new mutations are further facilitating their rapid evolution. And I think we're back down here, completing the circle, where actually water hemp is spreading globally now, and that these locally adapted populations are invading new regions in which where they're already perhaps at this fitness optimum in these intensified agricultural regimes. So I just want to thank you guys all for listening today. Thank my funding sources, mentors, and collaborators. And if any of this work interests you, I'd be really happy to hear from you and talk about some of the work we're planning to do in my lab at the University of Chicago. Thanks so much.